Welcome to Learn Chemistry Together. This video is about computational protein design and protein structure prediction. Overcoming decades of hindrance from slow experimental methods and vast theoretical conformations, recent breakthroughs have solved the protein folding grand challenge by enabling precise 3D structure predictions from amino acid sequences and a custom design of novel proteins. Background: after determination of the first protein structures, it was immediately recognized that protein tertiary, 3D, structures contain recurring regular so-called secondary structure elements, such as A helices and SS sheets, the orientation and packing of which, together with connecting loop regions, define the actual tertiary topology, Figure 1. Early secondary structure predictions, beginning with Linus Pauling's 1951 alpha helix model, and Cho and Fasman's propensity calculations, lacked high accuracy because they relied solely on 1D sequences while neglecting 3D tertiary interactions. At that time, the total number of experimentally determined structures in the protein data bank was also very modest, a few hundred, and it remained at this level until the 1990s, when the database really started to grow due to several advances in protein crystallography, Figure 2, insights into the hydrophobic effect, placing nonpolar side chains inward and polar groups outward while forming interior hydrogen bonds, inspired the earliest attempts at protein design. Based on this information, a perfect case for protein design would be amphiphilic helical structures, where one face of each A helix is hydrophilic, exposed to the solvent, and the other face is hydrophobic, which would allow packing with similarly hydrophobic surfaces in the interior of the design structure, Figure 3. Although biophysical principles enabled early successes like Reagan and DeGrado's 4-helix bundle and hex binary coded structures, these simple rules fail for complex atomic detail topologies, necessitating automated computational approaches. Computational protein design. In 1997, Dahiet and Mayo achieved the first fully computational de novo protein design by engineering a metal-free zinc finger variant, using a fixed backbone, dead-end elimination algorithm, Monte Carlo simulations, and empirical scoring to navigate sequence and side chain combinations. The resulting optimal design had 6 out of 28 residues in common with the original ZN finger sequence, 21% identity, and its 3D structure was determined by NMR spectroscopy, demonstrating a close resemblance to the computationally predicted structure, Figure 4. While Dahiet and Mayo's earlier work was restricted to fixed backbones, David Baker and colleagues achieved a major breakthrough in 2003 by designing and validating Top7, a novel 93 residue protein with fully optimized backbone conformations. This was a truly remarkable achievement for several reasons. First, it was a relatively large protein with 2 A helices and an SS sheet made up from 5 SS strands, and its predicted structure was in agreement with the experimental one, including the detailed side chain positions, Figure 5. Second, the author sought to design a folding pattern not found for any globular protein in the protein data bank. And third, the final Top7 design had no significant sequence similarity to any naturally occurring protein in the sequence databases. David Baker's 1999 Rosetta software revolutionized protein design to yield novel structures like Top7, while protein structure prediction advanced slowly through CASP experiments until machine learning analyses transformed the field by 2016. Protein structure prediction and AlphaFold. CASP-13 in 2018 marked a major breakthrough, as deep learning and convolutional neural networks translated distance maps into 3D structures. Capitalizing on this, DeepMind developed AlphaFold AF1, which used sequence alignments and protein data bank data to predict amino acid distance distributions, employing gradient descent to generate accurate 3D protein structures. With deep learning entering the structure prediction field, the performance had now risen to a GDT score of about 60%, Figure 6. and the AlphaFold team was clearly ahead of other participants. In 2018, Hasabi and his team dominated the Go and chess communities by publishing AlphaZero, a deep learning-based program that demonstrated unsurpassed performance in those two-party games. Though protein structure prediction still required further development to achieve near-experimental accuracy. AlphaFold 2, the real breakthrough. At CASP 14 in 2020, DeepMind's AlphaFold 2, developed by John Jumper and Demis Hassabis, achieved a historic breakthrough by matching experimental accuracy on difficult targets through an end-to-end transformer architecture with attention mechanisms. The AF2 network has two main blocks called the EvaFormer and the structure module, Figure 7. AlphaFold 2's architecture relies on two main components. The EvaFormer, which simultaneously updates multiple sequence alignments and pair representations, and the structure module, which uses invariant point attention to move residue triangles as rigid bodies into final 3D coordinates. This approach goes beyond mere pattern recognition, effectively learning the physical free energy surface of proteins. With both structural prediction and design largely solved, AlphaFold 2's creators mapped over 200 million protein structures in the UniProt database, expanding available structural data by orders of magnitude. Built upon decades of experimental data in the Protein Data Bank, the monumental achievements of David Baker, Demis Hassabis, and John Jumper have ushered in a transformative new era for biomedical research, synthetic biology, and targeted protein design. Thank you for watching. Click follow or subscribe for supporting this channel.